Okay, so before the real session and all the magic begins, I'm going to give a brief introduction to the Genome Editing Research Group. I am one of the co-chairs, Molly's one of the other ones, Shondra, as featured speaker in this session, is one. Guru, who just joined us, is also in our group. So we are a fairly small group. We do a lot of different technologies in our co-facilities. We range from a very small, very good high throughput co-facility to very, very small, like 3 people doing this stuff. And we do all ranges in between. The one thing we have in common is we all do genome editing, and we're trying to find, and we're working together to come up with the best practices for genome editing in co-facilities that we can then tell you so you can do it efficiently in your co's and really help out people at your institutions with the best edited models that you can get and functional screens. So scan the QR code if you're interested in joining the Genome Editing Research Group. We're planning on trying to decide what our study for the next year will be. And yes, if you are interested in joining, you can email me or Molly, and we will let you know how that happens. Thank you. Oh, yes, I forgot. Thank you. So it's not really a genome editing session tomorrow, but we as a group have put on the now infamously titled, I think, It Didn't Work, So I'm Not Paying. That is our session. And it is going to be all about the unique challenges faced by personnel-based codes that some other co-facilities don't really necessarily have to deal with. Different challenges that we have, including the title. Yes, that was born out of frustration. I can tell you all about it when I introduce the session tomorrow. So it's not a genome editing thing, so it's got broad applicability to everyone, even though it is us that's putting it on. Okay, thank you.
Let me know if you can't hear me. It's okay? I'm Jeremy, and I'm going to introduce this session really quickly. So this will be me and Shondra, who was kind of just introduced, from St. Jude, and also Kendall Condon from Bristol Myers Squibb. We're going to be talking about a pretty broad swath of genome engineering and from kind of different perspectives. And then I'm hoping to save as much time as possible for the end. So the way I engineered my talk after kind of seeing the audience here and what people might be interested in is to really briefly go over what we do. And then you can ask me specific questions if you're interested in like doing one of these things rather than to dive into one of them. So with that said, I'll get started. And I think we'll try to save questions to the end, unless something's confusing, then please let me know. So just some quick disclosures. I'm a consultant for BioHub and on the SAB of a company that is not related to genome engineering. So just one thing of nomenclature to get started that I'm going to use, and other people, it might not be universal. So genome engineering I'm going to use is like the really broad term to mean all the ways that we do insertions and deletions or even epigenetic modifications. So I'm using this broad definition. And then there are more specific uses like gene editing. Usually we refer to, that's like making a clonal cell line. So just to, so that people are on the same page as me here. And what we, so I'm the director of a platform called Advanced Technologies for Genomics CoLab at Columbia, and it is kind of a conglomeration of genomic technologies. And that includes genome engineering services, some spatial profiling that I'm not going to talk about, and some arrayed functional genomics. And this, although it's not directly related to pooled CRISPR screens, which is going to be my focus today, I did want to mention this because I think if you're into functional genomics, there are some like new opportunities that have just come up in the last maybe 6 months, particularly with 10x Genomics' Flex version 2 technology, which is called Apex. So I wanted to mention this really quickly because you might find it useful, and you can ask me more about it at the end. So my predecessor at Columbia invented something called PlateSeq, which was published in 2017, quite a long time ago in genomics years. It's probably 200 years ago in genomics time. That is a cheap way of doing bulk RNA-seq in a RAID format. And there's now we have an updated version, which is single-cell PlateSeq. And that is using basically a liquid handling automation system that does single-cell RNA sequencing in an arrayed format using 10x Genomics' Flex version 2 technology. So that has like barcodes that go in each well. All the RNA that you are quantifying in each cell in each well will get that. So you get single-cell data and arrayed data. So you could do compound screens, siRNA, cytokines. And this is the reason I threw this in here is there's a weird opportunity for cores because 10x Flex 2 has extremely high cost up front. It's like $25,000 a plate or $15,000 a plate if you're buying in packs of 4. And that's just for the base reagents, but you can split that up into reactions. So if you're a functional genomics arrayed person working with a friend who's a genomics person, then this it kind of presents an opportunity where you can do something that your the scientists you're working with can't do, because the really amazing thing is typically with 10x the reagents expire quickly enough that you can't just gamble $60,000. In this case, the limiting reagent you can buy separately. It's expensive, but it'll last for years because it's basically DNA. So this means that you can do some of the things I'm talking about today, you'll be able to do on a smaller scale and not just tens of millions of cells per week or whatever. If this seems interesting, then let me know and we'll bring it up at the end. But mostly what I wanted to talk about was pooled CRISPR technologies. And so for those of you who have gotten rusty on this, since it's like are we almost 10 years since the Nobel Prize now? It started out as a bacterial immune system. If you look at the left side of this diagram, this is showing the bacterial system, and it was a way that a bacteria could have a kind of learned immunity by putting bits of bacteriophage invading genetic material into their genome, and then pulling it out and basically having complexes of proteins floating around that would look for that genetic material, and then when they found it, a nuclease domain would then cut it, right? And so that's where we get the clustered regularly interspersed short palindromic repeats named for CRISPR. And that system was then turned into an engineering system by Jennifer Doudna and Emmanuel Charpentier. And we have hundreds and hundreds of variant technologies that have come out over the years. I think there are more than 200 base editors alone at this point. So this has become a massive field. There are so many CRISPR or CRISPR-like systems in nature, in archaea and bacteria. I think that when they trained an AI model, they trained on about 1 million. So Profluent was the company that did this. So they could actually create new ones de novo because there's so many out there. So this is a very widely used system in nature. And it has been used in research, which is what I'm mostly talking about, but also in therapeutics. And there, of course, we all know CRISPR therapeutics, the first being Casgevy in the United States, which is sickle cell anemia and beta thalassemia treatment. And there are a bunch more that are in clinical trials right now. In fact, this is a pretty old slide, but I think we were in the range of 70 clinical trials 2 years ago. And that's for a wide range of conditions. And this is, I would say, exponentially increasing right now. And part of that is because of base editing. Getting a therapeutic with a double-strand break is a bit tricky, especially as we've learned. There can be a lot of unexpected or unintended consequences. But once you get into epigenetic modification, because there are also clinical trials now for things like CRISPR-off or similar things, which basically are just methylating an area to turn off expression without any kind of break to DNA. And then base editing, which doesn't have to cause a double-strand break. Those are seen as a lot safer. So I think this is an exploding area. And although there are hundreds, like I said, hundreds of different CRISPR technologies out there, the ones that I'm just going to mention briefly are we do knockouts, and that's with Cas9 or Cas12a. I particularly like Cas12a. We use the Informer system, which is Informer, or Inzolia is the name of the library. So this is published by Trayvon Hart's group and John Dench's group at the Broad, and Trayvon's in Texas. This is a really fantastic tool because it's compact, because Cas12 has a superpower, which is that it can basically cleave out its own guides from an array. So you can have a compact all-in-one system and have really high efficiencies. This is what I recommend now for doing genome-wide screens. The Inzolia, you could buy it off Adgene for probably $1,000. And it not only covers the genome, but it also has paralogs. And so you can do paralogs for the first time. There's a ton of low-hanging fruit there. I think it's fantastic. So that library size is important because that is really critical to basically how the size of your screen is going to be. And in a lot of contexts now, if you're doing this in organoids or iPSC-derived cells, it's really hard to get enough cells to do your large screen. So CRISPR-I think most people are familiar with. So this is a catalytically dead Cas9. And so it doesn't cut anymore. You attach a repressor domain to it, and it will go to the promoter of a gene and silence transcription. It has the advantage of not having double-stranded break. Also, it can act pretty quickly because you just make a bunch and it binds there, and you're not waiting for multiple rounds of cell divisions for the indel to be successfully created biallelically. So CRISPR cutting, it's kind of like happening in one cell in this division and another cell in that. So it can take quite a while. This tends to be pretty quick. So we do a lot of like 4-day, 5-day. I've done it down as slow as 1 day with inducible systems. CRISPR-A is quite popular for smaller scale and is now getting better and better for larger screens. And this is where you attach activator domains and try to turn on transcription of a gene. For large pooled screens, I think we're now getting to a point where it's better. I think a few years ago it was not quite ready for that large scale because you would have to use mini guides, maybe 10 guides, just to turn on a single gene a bit. CRISPR-off is basically CRISPR-I, but with additional methylase domains attached to it so that you can methylate a promoter and have heritable repression. So that's a really cool trick where you can turn off a gene, then you could lose the Cas entirely in the daughter cells and granddaughter cells, but still have repression. And base editing, which Kendall will get into, is what it sounds like, where we have deaminases that are fused to Cas9, and the deamination is a trick we use to get the cell to make a mistake in correcting or attempting to correct that deamination and lead to a conversion. And so that's a way that we can make targeted base conversion. So I'll let her talk about it more, but it's one of the coolest tools if you're in a more translational or pharma side and you want to know, for example, what are the mutations that might confer resistance to this new drug. This is amazing for doing that. So I've rarely seen a technology that out of the box works so well. So to go more to a kind of core facility management side on this is how we operate. So the blue is the stuff that typically we are doing. So the left side of this is kind of showing the scale of large phenotypic CRISPR screens. This is how we buy our plastics, even just for 1 or 2 screens. It's massive scale. And that's because a lot of times if people you're working with might not have done the math. I did it here really quickly just to show if you're doing 20,000 genes in the genome with 3 guides times 1,000x coverage, and then you're doing a couple of replicates of a few conditions, you end up with a couple of billion cells really quickly. And 500 are being cultured. So it can become massive scale. It's not trivial, and it's something that I would say even people really experienced at cell culture tend to fail out the first time they do it, just because it's more of a project management issue than a cell culture issue. Kendall also is a pro at this, so she might talk about that. She, especially at flow sorting screens. this is our workflow. We are doing a lot of design and synthesis of the libraries, validating them, cloning and sequencing. So this is a custom library, not like a genome-wide one you could buy off Adgene. And then lentiviral production, and we functionally validate lentivirus in a standard cell line like A549, just to make sure when you give it to somebody, you're confident your virus is good. So otherwise, you will have a hard time knowing whether their cell line is difficult or your virus. If you're doing a custom screen, meaning only part of the genome, like if they want to do just their 50 genes they care about, then you have to do all of this each time. That's quite expensive because there's an NGS step in here you always will have to do. You'll always have to sequence the library to know the uniformity. Otherwise, you won't know the abundance of each guide. So I really push people towards genome-wide because it's actually, I charge the same amount for genome-wide as 50 genes from our point of view, because it's just the amount of labor involved in doing this well is quite a lot. Typically, the person we're collaborating with is doing the screen itself, which is cell line optimization, which is a lot of work. It could take a month. The screen and then guide RNA, they'll do a genomic DNA prep and give it to us, and we'll do QC and library preps. It's also good to work with somebody who does liquid handling automation for this. If it's a large screen, you'll be in multiple 96-well plates, and they have to keep getting spawned off into others because you're doing like milligrams of DNA. As Matt here knows, polymerase cannot handle having a milligram of DNA in a single reaction. It will quit. And so you will have to split things up into a lot of reactions. So it's good to have a friend who has a liquid handling robot. And then at the end, the sequencing. So that's kind of the workflow that we use. And I will go really briefly through the rest here. We are doing now a lot more perturb-seq. So instead of doing a viability readout, a flow sort readout, or that kind of traditional readout that has basically a bit of information, 2 bits of information, like live, dead, on, off, we are now switching more and more to perturb-seq. And because there have been these just tremendous drops in cost, both of sequencing and of the single cell library generation, using whatever technology you're using, we typically are using 10x, but I've used other ones. We've gotten to the point where now I'm getting fewer and fewer people who are wanting to do phenotypic screens. It doesn't change very much each year, but we've had an exponential increase in the amount of perturb-seq that we're doing. And perturb-seq was a bunch of papers published in 2016 to 17, and it basically involves putting your guide RNAs into individual cells, and then we're doing single-cell RNA sequencing where you also capture the RNA, and there's a bunch of different ways of capturing the guide. And but in the modern day, you can basically directly capture the guide either with probes, which is what we typically do now using 10x Flex, which is a probe-based system. That means you're not capturing mRNA. You have a probe for each. You have multiple probes for each RNA target. They bind and they get ligated together, and then you can amplify off of them, and that allows you to quantify the RNA. So that's what we do, but there are a bunch of methods. But the point is, you know what got knocked down or knocked out in each cell, and you get the full transcriptome data. We sometimes use a system where there are 2 guides in each cell, so you could knock down 2 genes, or if you're not sure which guide, how good your guides are, you could try 2. We don't need that quite as much anymore because we're better and better at guides. But I wanted to kind of finish off here by giving you the design considerations that we use. So the readout could be either, as I was just saying, a traditional live cells going to a 10x chromium method. So that would be like their 5' technology, or something they call multiome if you want to have a TAC-seq data at the same time, or the Flex V2, which is now overwhelmingly what I'm using for cost reasons. And part of it is because now you can kind of divide the cost up if you're doing massive experiments or small experiments and get better costs each way. And if you have questions, I can tell you how we do that. There are reasons you don't want to use it because it is a probe-based system, so it can't tell you certain things. And it's particularly not good if you have someone come to you and they have a very precious sample with only a few cells, which is very common with tumor samples because Flex is incredibly lossy. You will lose 50% if you're having a good day, and on a bad day, it could be a lot worse. So you don't want to have someone come with 2,000 precious tumor cells. There are some considerations with probe design, but I'm not going to get into that. And also, we have validated Flex, but now at this point in time, so have many other labs, and there are a ton of publications, including genome-wide publications, showing how well it works. And I'm going to close by saying a really important consideration in terms of cost is guide assignment. Guide assignment is not 100%. It's between 50 and, say, 90%, depending on the technology you're using. And that changes your price a lot, because it could be that you're throwing away half your cells because you couldn't tell what guide was in them. There's a paper called CRISPR, and it really helps you go through this. So I recommend looking at that if you're doing this and figuring out which method is optimal. So with that, I'm going to conclude and hand it over to Kendall, who's going to talk about base editing.
All right, hi everyone. As Jeremy mentioned, I'm Kendall Condon from Bristol-Myers Squibb, and I'm part of the functional genomics group there, and we support all aspects of functional genomics as it pertains to small molecule drug discovery. So today I'll go through a little vignette or case study where we used mutagenesis base editing to identify the binding site for our molecule BMS 986408. Now, for a little bit of background, immune checkpoint inhibitors have really transformed cancer therapies, but there remains hurdles in terms of primary resistance as well as acquired resistance. And so one strategy here is to look for differentiated mechanisms of action to pair with these PD-1 or CTLA-4 mechanisms. And one way that we attempted this was to use a phenotypic screening strategy to identify a small molecule that overcame the PD-1 mediated suppression. And so if you're interested in the chemistry behind how we went from our HTS hit, which was a DGK alpha inhibitor, to the current molecule that I'm studying today, you can look at several of these publications. And ultimately, we ended up with a dual DGK alpha, DGK zeta inhibitor. But it had a very interesting phenotype that I'll go through. And one question that was really left after we had designed this molecule and did a lot of SAR was, where does BMS 986408 actually bind to DGK alpha and DGK zeta? And I'll get into a little bit about why this was such a crucial question later on in the talk. But to give a little bit of background of what the DGKs are, they are diacylglycerol kinases that act at the plasma membrane to convert DAG into phosphatidic acid. And essentially these inhibitors of DGKs essentially turn off the brakes on T cells. And so that unleashes them to be better tumor cell killers, as well as they have specific increased effects on T cell activation. So they were a really exciting target to work on. Now, as a part of the functional genomics group, we kind of take on all aspects, and so all aspects of screening. And so what was very exciting about this molecule is that we actually knew a ton about it from the get-go. So we knew that these molecules bound directly to DGK alpha and DGK zeta from some cellular binding assays that are seen at that number 1 next to direct binding. And then additionally, these compounds also had this interesting phenotype of translocation, where they drove translocation of DGK alpha and DGK zeta from the cytosol to the plasma membrane. And then once at the plasma membrane, we were able to observe the inhibition of cellular DGK lipid kinase activity directly. And then finally, what was very interesting and what we were able to then leverage for our screening technologies, or for our screening strategy, was this phenotype of DGK alpha and DGK zeta degradation. And so what the biology team came to us with was this key question of, where does the compound engage the target? And this was really crucial because, we always like to run SAR and better understand our molecules to be able to design better molecules. And one way we do that is through looking at how the molecule actually binds the target directly. Now, this became a real challenge because there are no currently solved experimental structures for both full length or any of the human DGK alpha, DGK zeta. And so one way to do this as a functional genomics core was to kind of take a genetic look at this. And so what we did was use a endogenously tagged DGK alpha cell line that we generated in Jurkat cells. And that's what I'm showing here. Let me get my pointer. That's what I'm showing in this flow plot. We're looking at a histogram. So when we treat with our compound 408, we then see a decrease in the DGK alpha signal. And this was also verified with additional inhibitors. So we knew that this was proteasome dependent, and we could validate this cell line through looking at some of the other characterization that we had seen with this molecule previously. Now, getting into how we were going to take a genetic look at this or look at this through a genetic lens, we kind of considered 3 different techniques. And in general, deep mutational scanning is a really powerful technology, but is often difficult to execute quickly. And we're all on very tight timelines, as I'm sure all of you know. And so, we were a little bit scared away from this potentially because of the time that's associated with generating these libraries, as well as the deconvolution associated. And these are also quite expensive experiments to run. And then CRISPR tiling is nice because it gives you the resolution of a domain level, while also being a little bit easier to execute. But we needed more amino acid level resolution to be able to identify where our binding site was on our DGK. So we ultimately landed on CRISPR base editing. And to be very specific, we use this adenine base editor, which Jeremy mentioned previously. He did a lot of the work here, so I'm just going to go over that. We have these adenine base editors that generate these point mutations at a nucleotide resolution. And so ultimately, we chose this because of the relatively short timeline and the ease of generating libraries, although there is the caveat that this is not all by all point mutations, right? It's not deep mutational scanning, so that's a caveat here. And then a crucial part of this also was to generate or better understand what our coverage of these libraries would be. So we did this ahead of time and know that about 74% of DGK alpha and 74% of DGK alpha and 69% of DGK zeta can be targeted by the NG version of this base editor. All right, so with our libraries in hand, we went ahead and executed this FACS-based screen. So we take our endogenously tagged DGK alpha cell line, introduce our tiling library, and this was an all-in-one vector, which is, I think, really nice in this circumstance because you can add it into many other cell lines if you're interested in, different effects across different cell lines. Here we were mostly interested in Jurkat's or T cells, or T cell model. And what we can evaluate here are that there are cells that potentially have lethal mutations. We don't expect that from DGK alpha, but as a part of base editors, we include essential gene controls. And so when those essential gene controls drop out, we can then assess the quality of base editing in our screen overall. But once we have this viable mutant pool, we can then treat with our compound BMS-408 and then sort the high or resistant 10% of that population. And here we expect to identify point mutations that prevent DGK alpha induced or compound-induced DGK alpha degradation. And initially, when we were using this technology, we thought that it was very important to make sure we got a highly enriched cell pool. And so we actually did this iteratively. We did live cell sort, expanded those cells, retreated with compound, and then sorted again. And we can talk about later, if you guys are interested, if that's really needed, I think. The answer is probably not, potentially. Because what we saw when we looked at the QC of these screens was that we had really high correlation between replicates for both DGK alpha and DGK zeta, which maybe isn't a surprise. This is a very strong enrichment of a positive selection screen. But what was really nice was that we also saw, and this is looking at this plot on the right, you can see evidence for essential gene dropout. So if you look at the essential genes, you see that the points are below the 0 line, while guide RNAs that are targeting the no site or intergenic control center mostly around 0, indicating that we weren't bottlenecking significantly our library and that most of those mutations, as we expect, wouldn't cause an effect on viability at all. But then what was really, really exciting was actually this data, kind of set up like this, where we're looking at the domains of DGK alpha and DGK zeta across the x-axis, and then the log-fold change of our compound treated versus DMSO treated controls. And so this log-fold change is our treated controlled sorted population or fraction. And so what you can see really nicely, and what I've highlighted in the green dots, are mutations or residues that we identified between the two that localize in these kinase domains. So what was also very exciting is that we had kind of two almost like biological replicates, right, comparing DGK alpha to DGK zeta, and that allowed us to better have more confidence that what we were seeing was correct. But the question becomes, like, what do you go forward with? So what do you validate out of these screens? And if you're familiar with base editing, you can have bystander effects, you can have other issues with this data that makes deconvolution a little bit more difficult. It's not as easy as these are the guide RNAs, this is the gene, the follow-up validation is a knockout. So one strategy that we used was to actually use our guide RNA counting, which is what you typically do, but also couple that with visualizing these guide RNAs just on the gene body to see where you had hotspots. And so what you can see here is that when we overlap all this data, we see very nice hotspots exactly where I showed you on those kinase domains. And we coupled this with amplicon sequencing, so directly sequencing our enriched population at the DNA level to be able to identify which point mutations were actually being generated. So with base editing, you can have, as I mentioned, bystander effects. And so if those are what's driving your phenotype, you want to know about that before you go ahead and generate your endogenously tagged or your endogenous point mutations. So here we sequenced and then used CRISPR-Dav, which is our CRISPR data visualization software, to be able to really come up with a list of high priority mutants for endogenous knock-in. And I will say that the endogenous knock-in is quite useful and critical here because you want to maintain your screen is in an endogenous context. So you want to maintain that endogenous regulation of your gene while you validate. And what we showed really nicely was that all of the point mutations that we had picked out from our list using those two methods to kind of get a Venn diagram of what we wanted to go after, we found that these did prevent DGK alpha degradation. So what I'm showing here are just flow plots. As we saw before, when you treat with compound in the wild type setting, you then see a decrease in DGK alpha signal. But when we make this point mutation of H606R in these two clones, you no longer see that degradation phenotype. And then on the right, we can then quantify this and look at this as a mean fluorescence intensity and see that each one of these point mutations we identified prevents DGK alpha and DGK zeta degradation. And so I'll also mention that each point here represents a single cell clone from the population to eliminate potential clonal effects that we could have been seeing. And then finally, to truly convince ourselves that this was an effect that we were seeing on the target engagement side, we used cellular thermal shift assays, or CETSA. And the way that this works is that you have cells, and then you subject them to both compound treatment and untreated samples, and then you have a gradient of temperature, right? So you're looking for a destabilization or a stabilization of that temperature melting curve. And so what we observed is that in our wild type setting, we saw the dose-dependent response that we had seen before and expected. And then when we generated these point mutations, we no longer saw the dose dependence here, indicating that most likely these point mutations are preventing compound binding. Now, finally, as I mentioned, there are no structures of DGK alpha and DGK zeta, but everybody loves a structure. And so with AlphaFold, we can then generate those structures. That being said, obviously, the predictions potentially have caveats of maybe not being, fully trustable, right? Like, we don't know exactly. There's potentially lower confidence in some areas of these proteins. But very nicely, what we saw was that when we overlapped or highlighted where these point mutations were, we saw that the green dots, which were validated previously in our slide, really nicely sit in this pocket here. And then additionally, all of the orange residues were enriched, but we didn't go after for validation, and those also localize in the same area. And what's pretty interesting to me is that there's only 39% sequence overlap between these two domains, but we were able to really nicely identify both pockets in both DGK alpha and DGK zeta. And these also have different DAG substrate preferences. And just to go a little bit further into this on the structure side, again, the lighting or the coloring scheme is the same. The green is going to be validated residues and orange are going to be enriched residues. But as you can see very nicely, there are potentially predicted interactions between those enriched point mutations as well as some of the orange point mutations that really line the pocket where we have modeled in our 408 compound. And what was also very interesting here is that there was some other data that was unpublished that showed direct or potential suggested competitive binding with the DAG substrate. And so what was nice here is that this was the first time potentially that a DAG binding site had been identified in DGK alpha and DGK zeta. So this was like suggested, but interesting novel biology that we potentially uncovered. So overall, we use base editing to identify resistant point mutations for this degradation phenotype, as well as did functional validation for these degradation resistance phenotypes using SETSA and target engagement experiments. And then finally, the whole picture kind of came together when we integrated structural models, specifically using AlphaFold or more predictive modeling. And then with that, I'd just like to acknowledge a lot of folks that were part of the biology program, and then also a special thanks to other cores which were part of the Broad Institute, which helped in some of the library generation. Thanks.
All right, so I'm going to switch gears a little bit. I didn't realize you called genome editing was cell clonal stuff, and genome engineering is, you're saying screening? Okay. Okay. Because I've used them interchangeably, so I might do that, so I didn't adhere to that, but I will try my best. But I'm going to take you back to basically I'll tell you about the Center for Advanced Genome Engineering, which I direct at St. Jude Children's Research Hospital in Memphis, Tennessee. To start off, I just wanted to kind of tell you the services that we provide as a shared resource. We've been in existence for 9 years, and we provide 7 major services. So the first is we design, assemble, and validate guide RNAs. Now, we typically deliver our guide RNAs as RNPs, so like synthetic guide RNA with Cas9 protein, and that's what we put into cells. But we do work sometimes where we have to clone them into lentiviral vectors, or even adeno-associated vectors. So we have a menu of different vectors that we can clone into. We also create custom clonal cell lines, and this is where we spend most of our time. So people will come to us and they'll say, hey, I want to make this point mutation that I identified in my base editing screen, and I want to validate that as a hit. And we will go in there and make any modification in really any cell type. And so we create about 4 custom clonal cell lines every single week. So we do about 200 clonal cell lines every year. We also do edited cell pools. So we don't actually always need a clonal cell line. Sometimes with base editing or other technologies or even a knockout pool, maybe we just need to get a really highly edited pool, and that can be faster and cheaper for folks. So we create about 2 custom edited pools, which just means we have the needles in the haystack, but hopefully the needle's fairly large in terms of those pools. We also work with our animal facilities to generate custom animal models, and so we make about 1.5, it's not like half an animal, but you know what I mean, about 80 animal models. These are rodent models, typically mouse and rat, every year. We also do targeted next-generation sequencing or amplicon sequencing for CRISPR editing outcomes. So people can come to us at Cage, and if they're doing genome editing in their lab, they can basically throw their samples in. They can give us cell pellets, they can give us toe snips, they can give us tail snips, and we will do the targeted amplicon sequencing library prep, sequence it, and analyze their outcomes for them. And so within about 3.5 days, we'll be able to go from tail snip, toe snip, to their genome editing outcomes. And we do about 5,000 samples a week, which is about 10,000 PCRs, because we do a 2-step PCR process, which I'm not going to talk, well, actually I am going to talk about, sorry. And then we also facilitate high throughput genetic screens. Similar to what Jeremy was talking about, we do basically, I call it the buns of the sandwich. So we do the upfront design, assemble, validation of the library. They actually do the screen, and then we help them with, they give us back genomic DNA, and we do the sequencing prep for them, and then the initial analysis. So that's, they're the meat. Does that make sense? Maybe I shouldn't call it the buns, because maybe you're thinking about inappropriate things. Anyway, so we'll keep going. So the other thing that we offer is fitness assay. We call it a selfie assay. We published this, I think, last year. It's just a way of validating pooled CRISPR knockout screens, and it just tracks indel profiles over time. And I won't talk about that in much detail, but we do about 100 of those a year as well. So I did call it traditional genome editing or traditional genome engineering, and this is where we typically utilize genome editing, platforms like base editing, Cas12a, Cas9, prime editing we do some as well. But for our operational side of the genome editing facility, we use traditional, which means we are making a double-stranded break in the cell. The reason why we do this, and Kendall was mentioning this, is that base editing in particular, it has bystander effects. So if somebody says, hey, I want to make this A to G mutation, and we're like, okay, great, we made that A to G mutation, and we also got these bonus A to G mutations, they don't like that. And so with genome editing, the traditional genome editing approach, where we make a double-stranded break, we provide the cell with a donor template in which we have defined what the modification is going to be after repair. That is going to give us a more precise edit across an operational pipeline like ours, where we're doing a lot of different editing outcomes. Prime editing, I agree with you. It's not really, I make the joke, it's not ready for prime time yet. It is, if you have one site that you're going after and you can optimize it a lot, it's really great technology, but it requires quite a bit of optimization. And as you can see from our pipeline, what we're doing, we wouldn't have the ability to optimize like 80 different genomic targets every single week. So we rely on traditional genome editing. This is just making a targeted double-stranded break in genomic DNA, and then allowing the cell's endogenous repair pathways to repair that double-stranded break in such a way that we either get the knockout, which is through that non-homologous end-joining pathway over here on the left, or if we supply the cell with this extrachromosomal donor, which can either be a single-stranded oligo, can be AAV donor, it can be double-stranded plasmid, but basically something that tells the cell how we want that repair to happen. So we rely on this traditional genome editing approach. In terms of scope, in terms of how many sites have we done custom edits for, so not screens, this doesn't include our screen guide RNAs. This is just a fake karyotype, I guess, where you can see all the different chromosomes. These are just these are the mouse chromosomes, these are the human chromosomes, and these are the loci. Each of the little red dots or red lines are where we have actually made edits in a cell line or animal. So all this to say is we do a lot of genome editing, and we do it robustly, and so we have to we think about operationalizing things as best we can. And our operations really rely on genome editing with a targeted next-generation sequencing or amplicon-seq. It's really important for us to be able to do that targeted amplicon sequencing. So I make the joke that genome editors spend about 5% of their time doing genome editing and about 95% of their time seeing if we did what we said we were doing. And we do that using targeted next-generation sequencing or targeted amplicon sequencing. So that 5%, that doing the genome editing, is basically introducing the guide RNAs and the Cas9 or the Cas12a protein into cells. You let this in your donors if you're doing knock-ins. You let the cells do their work. If you're making an animal model, you would put that into a zygote, and you let the mom do all the work, which can I get a amen? No? I heard some dads laugh. I like that. Okay, thank you. So basically the genome editing happens once you introduce those reagents into the cells or the zygotes. Then you let the culturing happen. We're not really doing anything. We're just waiting on the cells to do their thing to repair those double-stranded breaks. And then what we do, where we spend a lot of our time, is we say, did we actually make that modification? And so we do this really briefly in a two-step process. First, we have a PCR primers that basically amplify the region of interest. We know where we're making that modification, so those primers land around that cut site. And then we have these common Illumina adapters here. These are those little tails. And that's PCR1, that's target specific. We're amplifying the region of interest. And then PCR number 2, we basically add on barcodes and the rest of the Illumina adapter so that we can actually tell, once we do all these 10,000 PCRs, we can put them all together, put them in one tube and run them on one flow cell, which makes it economical. We can deconvolute those based on those barcodes. So we know every barcode and where it came from based on how we indexed it. And so this pipeline takes us about 3.5 days from start to finish. So people at St. Jude, if you come to us and you give us a toe or a cell pellet, we can give you the results of your editing outcome, which means we'll take your genomic DNA or harvest your genomic DNA, we will amplify that region, we will index it, we will sequence it, we will analyze it, and we'll give you the results in about 3.5 days from Monday to Thursday. This is our shtick, right? So I spent a bit of time on it because we can do this really, really fast, and it's very, very cheap. It's basically free because we do so many samples in a run. All right, so I try to come up with, you've already laughed a couple times, so I'm gonna tell my husband I succeeded, because he usually says, do they laugh? And I usually say no. But today you've laughed a couple times, so it's gonna go. So here's my tales from the clone zone. Thank you. You win. I like you the most. Okay, so basically, I am not going to scare you, but I have been doing genome editing since 2003, before CRISPR was cool. And there are things that I'm still learning about clonal analysis and how do you make high-value clones. So I'm going to give you 3 tales from the crypt of Clone Zone. We're going to talk about low-coverage whole genome sequencing for making high-quality edited clones. I'm going to talk about barcoded donors or barcoding donors so they can assess clonal diversity and allelic representation. And if I have time, I'm going to talk about aberrant targeted integration, where the plasmid backbone actually integrates at the target site. And I think this is grossly reported. And you can see it occasionally in papers, but it is not really being looked at. And it's happening a lot in our hands in what we're seeing. So I'm going to try to talk about those 3 things. So the first story is going to be about using low-coverage whole genome sequencing. We call it LOCO because it's crazy. Yeah. You guys over here, you got to... Yeah, tough crowd. Okay. So Patrick Connolly in the lab is the one that has really been spearheading this, and we now offer this as a service, and I'll tell you how we got there. So as I told you, we do a lot of genome editing. We've made over 1,500 custom clonal genome-edited cell lines. Our genotyping is typically done with targeted amplicon sequencing, so we do a whole bunch of PCRs, we sequence it, we use short read, so we're limited to about 600 base pairs. And the things that we might miss, so here's happy Shondra holding DNA, and here's the primers. These red guys are the primers. The green stars are where we basically are going to make that modification. Anything that we do that into this genomic DNA that causes one of these primer sites to go away means that we're going to be blind to that allele with the way we're genotyping, right? So things like a large deletion, so we make a double-stranded break and it chews back and we lose a primer binding site. We will not pick that up in our genotyping. If you have a large chromosomal aberration, meaning that you have a translocation, we would not pick that up. If you have a SNP in the primer binding site, we would not pick that up. We have some QC from that, but we might miss that. If you have a random integration of the backbone of the donor plasmid, we would not pick that up. If you have concatamers and it causes those primers they're too big, you would not pick that up. So there's quite a few things that basically mean you could have things that you might think you have something and you don't, or you might have missed an allele because you aren't fully QCing your cell lines. So here's the case of, I think it's called DYRK1A. I don't know if that's actually what the lab calls it, DYRK1A. They came to us and they said, hey, we want to do a C-terminal tag of this gene. We said, great, we're going to do that for you. We designed a donor and a guide. Here's our donor. In this case, we were using 800 base pair homology arms. We typically use 400 now. They wanted a tag with a D tag or this degradation domain to degrade the protein. We put in a linker and a D tag in between. We designed all these little primers so that we could do our PCR-based genotyping. We do 3 PCRs typically. So we do something called a wild type or a zygosity PCR, which has primers that land inside the homology arms. But if you've gotten the correct integration, it basically causes the wild type sequence to go from, in this case, about 400 base pairs to about 900 base pairs. So if you have a homozygous clone, you get a PCR that's 400, that means you have a wild type allele remaining. If you get a PCR of only 900 base pairs, that means you have shifted those primers apart. We also do a 5' junction and a 3' junction. Oh, I have animation for this, sorry. 5' junction is basically has one primer that's genomic and one primer that's inside of the insert that you're putting in, so in this case the D tag. And you will only get, that's a yes/no answer. If you get it's positive, that means you got the integration at the targeted site. We also have the 3' junction, which is the same thing on the right-hand side, or on the 3' side. And then we do this zygosity PCR, where we're basically looking to see, do we have a homozygous clone? Because when we look at this amplicon, it basically shifts those primers out. So we did this. We did this in NOM6 cells. Everything looked good. Here's our 5' junction. We have a parental cell line, no band because we didn't have the integration. We have a no target in control. And then we have these two clones, and then that's our pool for a positive control. So everything looks good. We have a 5', 3'. We sequence these. They look beautiful. We did our zygosity test, and both of these clones only show that bigger band. So everything looks good. We hand it off to them. We're like, here you go. Here's your NOM6 cell. Sequence confirmed. We've done all these PCRs. Good luck to you. We only look at the DNA level. We are only using targeted amplicon sequencing. And we always tell the end user, you should functionally validate this with whatever assays you have at the RNA level and at the protein level. But they don't always do that. But this one did. And so what they did is they took this and they were performing RNA-seq, and they found that neither clone was actually homozygous for the edit that we thought, and they actually had an untagged RNA that was still being made. So we were like, that's interesting. And so they were actually able to look at the sequence of the RNA that was being made, and they had some ideas of like, hey, it looks like there's a larger indel. And so we said, well, why don't we use this as an opportunity to start using low-coverage whole genome sequencing? Sequencing keeps getting cheaper and cheaper, and so this was at this time in our lives in biomedical research, it is actually affordable-ish to do low-coverage whole genome sequencing. So why low-co WGS? One, it's about 10 to 16x coverage instead of the normal like 30, 32x coverage whenever you do typical whole genome sequencing. So it's cheaper. You're getting less reads, but for clones, you probably don't need 32x coverage. It's going to detect small and large deletions. It's going to detect larger chromosomal aberrations. All those things we were going to miss when we were looking at just the spot of interest, we can now pick up most of those with low-coverage whole genome sequencing. So we did it. Shondra's happy. Cartoon Shondra. I have no teeth. It's just like one tooth, but anyway. I just noticed that. Okay, so if you haven't ever looked at IGV, this is basically showing you what it looks like. So quick tutorial. This is sequencing reads, and the bigger the mountains are, the more reads you have. So for low coverage whole genome sequencing, you're going to get about 10 to 16, and so each one of these little guys, arrows underneath, is a read. So if it's 10, you would expect that for every little area, you have about 10 reads that are actually underneath that area, okay? And then this little thing right here, if it's rainbow colored, that means it didn't match back to the reference genome. And so this is good, right? We shoved in that FKBP, that D tag, and so where we shoved it in, which is right here, you would get this rainbow because it doesn't match, because it's not in the genome, the reference genome. All right, so here's our test case, this DIRC1A loco WGS test. So here's our parental. This is kind of typical. You see this like kind of this pile up. It's just kind of real even ebbs and flows, right? So sometimes you get better coverage than others. And here, this is actually a little bit kind of junky, a little bit bad sequencing quality, but you see these rainbow things, they're kind of all over the place. They're not right at one spot. When we looked at those two clones that were giving us problems, that they said, hey, this isn't actually a homozygous clone, these two clones here, we saw that instead of having this nice rolling landscape, we had drop-offs. We had cliffs or buttes. I don't know if that's a word. It is a word, right? Arizona has buttes. You know what I'm talking about? Yes, Arizona, yes, okay. You have buttes. This is a butte. It's not beautiful. Anyway, okay, sorry. I get it. I digress. Okay, so basically we saw these drop-offs in reads, and that was saying, hey, there's a problem here. We've lost coverage. And what that really went back to is if we looked at the sequence and we aligned the sequence, you can see this clone, 14D9, had an allele that was missing the sequence, and this clone, 15D3, was missing all of this sequence. So when we did our PCR, which had these primers, we missed this one because the primer binding site was gone. And when we did this one, we missed this primer wouldn't bind, so we missed that allele. So we thought it was homozygous because all the sequencing looked the same, but in fact, we had missed an allele. All right, so then, this is my, okay, thank you. She's already laughing. I love it. I haven't got to the, okay, but you see my little face in the glasses? Okay. So basically, we had detected this. We had already done about 1,500 custom clonal lines. Not all of them were homozygous knock-ins, but we were like, okay, how far down the rabbit hole do we really want to go? So I said, yeah, let's do it. Let's see what we get. And so there's Shondra taking the red pill. She's going down the rabbit hole. We decided we're going to look at 20 clones that were homozygous. We thought that they're homozygous from targeted amplicon sequencing. This was 8 genes, 9 different parental backgrounds. We did 11 different edits. Some were done with plasmid donors, some were done with SSODNs, and one was done with a PCR donor. And we just said, okay, what do we see? So here is an example of using a plasmid donor, and we had no unexpected deletions. So again, here's our parental. Sorry, it's kind of tiny. But the parental, you can see it's a nice rolling landscape. Here, again, you have a nice rolling landscape, and you see a solid drop at where the target site, the integration is. But when we look at the actual sequence reads, again, each one of these little gray arrows is a sequence read. We are just having those rainbow areas happening right at the gut site, which is the tag. So this looks good. So this is what we expect to see. We had no unexpected deletions. But we also found plenty of examples of where even when we were using an SSODN, where we actually saw the buttes, I think this is a butte over here, where we saw these drops in reads. And so here's an example of 2 different clones that we did using SSODN, and we were putting in a point mutation, which you can see the point mutation are those little red base pairs there. And so this clone we thought was homozygous, but in fact we had just missed alleles. So long story short, we basically analyzed these 20 clones. 5, so 25% actually contain large deletions that we had missed with our targeted amplicon sequencing. 2 were from the same project, so maybe you could say it's chromosomal or loci specific, maybe it's context specific. And I guess the good news is that most of the time the large deletions would have probably knocked out the untargeted allele. So like in the D tag, if it was on the N-terminal end, maybe you knock out one allele and you only have the D-tagged allele expressed. So maybe there's some good news. In this case, we basically had backup clones, so we could go back and give them backup clones, but they had done a lot of work on these clones, so we want to always think about how do we get the best clones possible. Summary of this portion of the talk, and I'm going to have to zoom through. I'm going to just do one other section. I'm not going to get to the last part, but if you want to talk about it at the talk, we can, the panel. Extra care should be taken whenever you're looking at homozygous clones because you might be missing something. You don't know what you're missing unless you look. For knockout clones, this is easier because we just look for unique indels. So we screen a whole bunch of clones, and if we feel like the indels are showing us that the clones are diploid or triploid, we just look for unique indels at those ratios, like 33, 33, 33. And then, as I mentioned, large deletions often knock out the gene, so you're only getting your modified allele expressed. So we all started offering loco WGS for homozygous clones. But how do we reduce the chances of missing that without having to do loco WGS, which we will do, but we thought other ways to make this better. And I think the answer is yes. And we started using barcoded donors. So this was done by Richard Bajpai in the lab. And we started using barcode donors because when we're looking at homozygous clones, the reads all look the same, as I just mentioned. So like this is an example of where we were putting in this 3X flag tag into this gene cereblon, CRBN1, or CRBN. 2 different clones, and when we do the NGS, they look the same. So I don't know, are they from the same parental cell? Are they homozygous? Am I missing something? So we can't really track the alleles. But if we were able to put in a barcode, a unique identifier into the genome, then we can track the alleles, and we can also make sure that it's an independent clone or of unique origin. So I just mentioned this. So here's our edited pool. Let's say we have 2 unique edits, and we deposit those into 96-well plates. Those will be unique snowflakes. That's what we want. However, if you have a cell and you get an edit in one, and then it divides, and now you have 3 cells, but they're all from the same parental cell, then you're getting subclones, and that's not ideal. When we're saying we're giving people 2 clones, we want them to be unique. So really quickly, you can measure diversity through barcoding. So in this example, a lab came to us. This is that DIRC1A again. We wanted to put a HYBIT tag in, and then we also put a barcode in after the stop codon. So we just used 8 base pairs that were randomized when we synthesized this SSODN donor. The donor had 60 base pairs of homology, flanking the cut site. It inserted our HYBIT tag, and then it also had this 8 base pair randomized sequence. We did a little bit of sequencing, so we just did 500x coverage, and we were able to see that we basically get all unique reads because there's basically 100,000 barcodes in here, and so we didn't see any. So that meant we had a lot of editing going on. We single cell sorted them 1 cell per well into a 96-well plate, and then we assessed what the clones look like. So in this case, we had a high diversity of clones. This was done in K562 cells. 45% of the clones actually had a barcode in. That means that they went through HDR. When we looked at the individual clones, 7% had multiple barcodes, meaning they had more than 1 barcode inside. 1% had only 1 barcode, and then 37% of the clones had had a barcode or codes and indels. So in K562 cells, there's actually 4 copies of this gene. So it wasn't that surprising to us that most of the cells had multiple barcodes. But we said, what about if we're looking at a diploid cell line? Would this look different? And so we took U2S cells, which are diploid for this gene, not completely diploid, in this case, this gene. And what we did see is that we saw more clones had only 1 barcode, which was a little bit concerning to us. And the reason why you would think, okay, if you might have only 1 barcode, because if you get one of those larger indels, now you could have 1 barcode and 1 missed allele. If you have 6 copies or 4 copies, the chances of you getting multiple barcodes is higher because you probably won't have large deletions on all of the alleles. So we were like, okay, we didn't have any duplicated barcodes across the clones, but what about those clones with 1 barcode? So we suspected that we were just missing those alleles because of our targeted amplicon sequencing. And so what you can hear, oh, it's teeny tiny, sorry. What you can see here is we just did a 2 kb PCR. So we just went out about 2 kb from that cut site, and then we did a 2 kb PCR, and we could see that if you got a dropout, see these bands, the lower bands show us that we were missing, we had a larger deletion that we were missing when we were doing the short targeted amplicon sequencing. And then we did a 5 kb PCR, and we saw yet another clone dropped out, and then we did a 10 kb PCR, and yet another clone grew out. So long story short is that the clones that had just one barcode, most of them contained a deletion as well. And so 9 out of the 12 actually contained a deletion that we confirmed with sequencing, Sanger sequencing, that they actually had a deletion. So yes, I'm going to stop. I'm going to show you one more slide, and then I'll sum it up, I promise. This is just another example, but it's another way, because I showed you the last example was a tag and a stop codon, and then we shoved in this random 8 base pair sequence. But you could actually do this in the coding sequence as well. And so this is what we have been doing more recently. In this case, the investigator came to us and they wanted to tag this MED13 gene, and they wanted to do it on the N-terminal end, and they wanted to put an alpha tag and a TEV binding site. So what we did is we put a GS, which is the flexible linker. A lot of times people will put these GS linkers in between like a GFP and a protein. And so all we did was we randomized the wobble position of those G's and those S's. And so that allowed us, so we just put, when we synthesized this SSOD in, we just said N instead of A or whatever. And that allowed us to have these barcodes that we can actually track all of the alleles. So in K562s, there's 3 copies of this gene, and we were able to uniquely identify all 3 of those by using these barcodes. I'm just showing you that's where it was. I'm showing you the wobble position here. S has, you can use, basically change to all 4 nucleotides, and so same thing with G. And I'll stop there and just wrap up and say that these barcodes allow us to see that there was a good diversity in the pools in the beginning. So it's not like we're getting one edit and then you're getting a whole bunch of daughter cells. We actually start with a whole bunch of edits, which was good. We did not see duplicate barcodes within different clones, but sometimes we saw like twice as many reads for one barcode as the other, which we think you get a cut on one allele and then it repairs off of a sister allele that already had a barcode. The barcodes have really helped us get better clones without having to do a whole bunch of extra work, so I highly recommend those. And then, most single barcoded clones contain larger deletions. So I think even just doing that larger 2 kb PCR as a QC check, it's super easy to do. That can actually make higher quality clones in the end. And that's what I mean by the out-out PCR. So I will wrap up there, and we will take questions.
Hello? Yeah.
Hi, thanks for the talks. That was great. Shondra, when you edit a cell line and you see a frame shift or a premature stop codon early on in the coding sequence, do you ever find examples where there's still some residual activity, like either domains retained or some scaffolding is retained, and you still don't see a phenotype from the knockout? Have you ever run across those examples?
I want to make sure I understand your question. So are you asking, whenever we try to knock out a gene using non-homologous end joining and you get this premature stop codon, we rely on nonsense mediated decay to degrade the protein?
Yeah.
Do we ever see protein left behind or activity of the protein?
Exactly.
I'm not the best person to answer that. For sure it's been reported because we don't often follow up with the clones, right? Like I'm not doing the Western blot. I can say that most people don't come back to us unless there's problems, and people haven't come back to us. Like the DIRC1A that came back to us because they said there's a problem. Certainly in the literature, it has been widely reported that, not widely, somewhat reported that you can, and we have seen exon skipping sometimes. So so basically you make an indel here. We target early conserved exons, but sometimes the cell will figure out a way of getting around it. I think it's more rare, but I think it has been reported. I'm not maybe the best person to answer that question, but because we don't follow up functionally on the knockouts. People aren't coming to us and saying, hey, there's protein here. If they do, usually it's a bad antibody, is what I would say. The first thing I say is check an antibody, and then they check it, and they're like, okay, it's gone. So I think it's not a huge problem, but I think it is something to think about. I don't know if you guys have thoughts on that.
I agree with it. It's the antibody's fault thing.
Most of the time.
Most of the time.
Yeah, we had, just to add a little context, I've only seen this happen once. You've done way more engineering than I have, and all the antibodies were the C-terminus, right? So and it was a little N-terminus we suspected was left over, but couldn't detect it. It looked like a clean knockout, right? It was a frame shift, and so, but there was still, it was complete, the functionality was completely retained. So, and we didn't know a whole lot about this protein.
Paralog though?
No,
No paralog?
Yeah, that's a good question. The disease expert did not say there was a paralog to think about, but we suspected that there was enough protein left to do what, whatever underlying function that was driving the disease of interest at the time, but had no way to prove it, right? So it was very frustrating. So I was hoping you're like, yeah, I've seen this 5 times and happens a lot, but no, pretty
I
rare.
would take your line and do a genome-wide screen in it to find the new homolog. It's a lot of work to prove that you did it right, that it was done right, but I
Yeah.
bet that there's another protein that was covering.
Yeah,
At least it's possible.
the
Yeah.
last I saw, they were going to go and flank it and literally chop out the entire loci to see if anything happened. I'm not sure how it turned out. So thank you.
Thanks for nothing, right?
Hi. So if you have you have to do array screening for 50, 100 genes, what do you do for validation? Do you do amplicon-seq, for example, for each well, or do you use just do some positive control? For pool screening.
Pool screening, I think.
Yeah.
So
Pool knockout screening.
so if you're doing like a phenotypic genome-wide screen or something,
No, not genome
Okay.
genome-wide. You have like 100 targets,
yeah.
and you want to do array, like each well has a single gene
Oh, array
target.
validation of
Yeah.
it.
So
Yeah.
how do you validate it? You do each well amplicon-seq, or
Yeah, that would be one way you could do it. If you had that many hits, there could be follow-up screens with a different technology sometimes. So sometimes we would do that. It kind of depends on what you think might be your confounds. So an arrayed amplicon-seq, a screen with a different technology. So like a CRISPR-I screen instead of a cutting screen if you're worried that the cutting was causing some other problem. Kendall?
Yeah, I mean, so when we've validated pooled screens with arrayed screening, we have gone through and done a validation process and done individual amplicon sequencing just to validate that the process, like the workflow, is working. And then in my experience, doing that individual amplicon sequencing across like a 96 well or a 384 well plate has just been too labor intensive. And I'd say that you can predict primers, but sometimes they fail. And I don't think a lot of folks have groups big enough to be optimizing the annealing temperature of 96 primers for one screen. So what we've done is, run what we have in terms of amplicon sequencing and then optimize the best that you can, but just go with that. And if there are genes that a group's particularly interested in and they say, okay, this is a high priority, then we go back, do the optimization, and make sure that those populations or those wells were completely edited. But now there's new arrayed technologies like that from some of the vendors that are probably here, where they've generated guide RNAs where you have multiple guide RNAs per well. So like increasing your likelihood that you're getting target gene disruption.
So 100's a lot. So I definitely recommend doing a follow-up screen to get it down to like a 20s, 30s-ish if you can. And then we published a paper, the selfie assay is just basically you do targeted amplicon sequencing, but you look at the indels over time. So if you're looking for, if you're doing knockout screens, you're probably looking for live dead. Is your phenotype, I'm guessing?
no, these are like novel targets, so we don't even know the phenotype. We are doing it in primary T cells.
But you have, for your screen, you're going to have a phenotype that you're looking for. And typically knockout is like, if I knock out this gene, does it kill the cells? And thus
Then we will do functional assay, but first, I don't expect them to all the cells die. So these probably will be viable cells that we will take and do some cytokine readout or something. But first, we want to validate if we knock out the gene that we thought we'd knock out, but in large scale.
yeah, I mean, Synthego or Editco or whatever they're called now, they do the 3 guides per well. I think that's a pretty good way. And you can get them for like $99 a well, I think.
Thank you.
I just want to point something out. You've made tons of custom pools, And you were saying that one of the biggest costs and challenges, Jerry, of your core is creating those pools. Have you guys ever thought of joining forces in that space? Because if you can only target the genes that are expressed in that cell, most cells only express up to, like, even cancer cells express like 8,000, right? And if you're just doing knockout, you really don't need to address non-expressed genes. So you could drastically reduce the coverage and scale of screens. With custom pools, it's just the pain in the butt to make them. Have you guys ever thought of collaborating or working together on that?
I mean, I think he's making custom pools.
Yeah, we make tons of
libraries.
custom pools. It's just like you can make a genome-wide once and amortize the cost.
Yeah.
And so that's typically what I recommend. Also, we rarely do cutting. We do pooled CRISPR-I screens 90% of the time. But yeah, it's just that the effort of any custom pool is almost the same as the effort of genome-wide.
And the biggest cost is the oligo pool, right? Like whenever you do custom, you have to order the oligo pool to clone in. That's like more than we charge just to the vendor.
Yeah, I think maybe, and if you're just screening in cancer cells, you can make as many as you need, right? But if you're moving to iPSC derived or primary cells, even then they become more precious and kind of limits your scale as well. But
yeah, so actually I'll say something on that too. So there's a paper that's gonna come out in Nature Methods by a group at Stanford, Michael McManus's group, and they're gonna argue that we can reduce the number in those. So what he's saying is there are a lot of times when we can't just grow a ton of cells and it's hard to grow and differentiate. So they're arguing for high MOI screening. And the compressed perturb-seq paper came out like a year or two ago arguing the same thing. And the field of pooled CRISPR screening, I feel, is moving now to we can do higher MOI, meaning having more than one guide per cell, and then deconvolving the readout. And there are now multiple papers on this. So I think genome-wide, yeah, if you're differentiating into something that you can really only get a few 100,000 of, but I think now we'll get to the point where we don't need that many cells for hopefully for genome-wide.
yeah, and I think if really, and we published on this a few years ago, if you just improve the quality and guide representation of the pools, you can drastically reduce your coverage because you don't have those extremes where you have 1 million of one guide and 0 of a bunch of others. I'll add one thing to that high MOI because this probably won't ever be published, but we had done some CRISPR QTL work, right, based on Molly Gasparini's use of CRISPR-I to address non-coding regions. So interestingly, you can silence potential regulatory elements with CRISPR-I, and because she's using it to survey a bunch of SNPs and she's not sure if they're actually in regulatory elements, she did like an MOI of, I don't remember, it was high, right? I think when we replicated it, we did 20, and what we saw is after about 5 integrations, all you started seeing was a viral response from the cells, and we saw this in, I think, primary kidney cells and a couple of immortalized cell lines. And so we kept seeing the same genes show up again and again, and we were like, oh, what is this? And then we kind of, and so like I said, we hadn't really nailed it down, and I'd love to publish on this, but you basically lost the kind of the response of interest, and it all became a viral response.
We can also put multiple guides in one virus too, though. And now there are ways like tRNA arrays where you could put a lot of guides into a single. So I agree with you, but I think there are tricks to get around that too.
Yeah, and I think it'd be so much more efficient if you could just do high MOI. I've been wanting to do something like that for a long time, so I think it's cool. Yeah. All right, I'll let other people talk.
Everybody's rushing to the microphone.
Great presentation, thank you. About your large donors, is it AV or single strand?
Yeah, so we in cell lines, we typically use double-stranded DNA, just plasmids, but I was just about to show you that that's bad. And so we've moved to AAV donors.
AV.
And we use SSODNs if it's less than 200 base pair total.
Right,
like 120.
because I had some people use up to 2,000 and those.
Yeah, I
doesn't
think, and
degrade.
Guru can speak to this too, but like IDT stopped even making things that were big because
wouldn't it
synthesis
degrade
errors,
or
right?
efficiency go
Yeah,
down?
and so like with mice, we were using ssDNAs to make mice, but what would happen is we would see non-related to the cut site, we would see mutations that were introduced during synthesis of the donors. And so we basically have moved to, we use SSODNs, short, you don't get very many synthesis errors. The longer you get, the more likely you're going to get it. So we just moved to AAVs. And we're getting much higher rates of knock-in with AAVs, probably because you get more copies with the ITRs.
And your hybrid tagging donor, it's
That
like
was
16,
SSOD.
6.
Yeah, we usually use 40 base pair homology arms, so we can get about
Oh.
120 base pair insert if you're right at the cut site.
That's enough, 40, 60.
Yeah, 40. Yeah, I think IDT says like do 34 to 36 or something, but we usually just do 40 because it's easier math.
I see. And our core in your Mount Sinai stem cell engineering core.
Yeah.
So we get orders from our users like 2-3 mutations, right? Yeah. Something like that. And how do you get customers like for pulling so many mutations, right?
Yeah, so I mean, if you're lucky enough it's a base edit and it doesn't have a bystander, you could do base editing multiplex, right? But it's rare that you can actually do that because there's almost always something by that's going to cause a problem. So we do it sequentially. For iPSCs, the editing rate for point mutations is quite good, 20-30% oftentimes.
Right,
And
right.
so we would do the first edit, we would wait a week, we would put in the reagents for the second edit, we'd wait a week, we'd put the reagents in for the third edit, and we'd wait a week, and then we would screen the clones. We never try to make 2 cuts at the same time because then you get translocations or deletions.
Right.
But we don't want to screen everything because it takes forever to do that. So we try to get as much, and we check the editing at each of those sites. And as long as you're like in the 20-30% range, you can likely get all 3. If you don't, maybe you just get 2, and then you have to stack on the third. Or sometimes you're really unlucky and you have to do it sequentially, like 3 unique cell lines. We charge for whatever we do. So if we actually have to make 3 unique clonal cell lines, they have to pay for 3 unique clonal cell lines. They don't love that, but.
And the last question, how do you address the off-target screening?
And the screen.
Off-targets.
And the clones are in the
Off-targets.
in clones or in
In
Old
clones,
screen.
yes, in clones.
So we basically try to design our guides well. So we try to design our guides to have at least 3 base pairs of mismatched to any other site in the genome. And early on in 2014, there was a paper from Versailles. I'm probably saying that wrong. That if you look at clones that have targeted edits in them, the chances of having an off-target event if they have 3 base pairs of mismatched to any other site in the genome is very, very low. So the chances of having an off-target if you have, you're going to put in 10 million cells into a patient is very, very high. But if you're looking at uniquely edited clones, the chances are low if you've designed them well. And then if we try to give them 2 or 3 clones, right? So we basically don't look at off-targets in terms of like small indels. We say we're going to design the guide well. We're going to give you at least 2 clones if we can, 3 clones if you want them. And if they're still really, really worried about it, they can do whole genome sequencing. It's very hard to look at indels. It's very hard to actually find indels that are not related to the cut site. And if they're very, very worried about it, and I have never had somebody take me up on this, design 2 different unique guides, make the same edit using 2 different guides, and then the chances of having the same off-target are even lower. But they'd have to pay for 2 cell lines, and they don't usually do that. They're not that worried.
Reassuring. Thank you.
I think we're at time, but if anyone has questions, you can run up after. But thank you.